Okay, so first off, we have apoptosis and cancer cells. Apoptosis is when cells die off naturally. It's a function that's naturally built into the cell life. It can also happen like during embryos, embryonic stages, so that you don't have the webbing between your toes, that those cells there will automatically die off before the baby's born. Cancer, on the other hand, is whenever cells start to divide uncontrollably and very rapidly. Two of the ways that we actually treat cancer, which is with chemotherapy and radiation, actually will force those mutated cells to go into apoptosis, so they die off, so our body can be rid of the cancer. So our first little diagram is of the cell life. See if I can get this in here. And so over here on the side, we have what we call our G0 stage. That is where our cells spend most of their time, is in the G0 stage. That's just when they're performing their basic functions that they're meant to do. Next we have our G1 stage, which is when the cells actually begin to grow. Then we have our S stage, which is when the DNA synthesizes. We have our G2 stage, which is when the cells prepare to actually divide. And then finally we go into mitosis, which is when the cells actually divide. The G1, S, and G2 stages are all known as interphases. Mitosis and the G0 stages are separate things. Set that one off to the side. So looking a little bit deeper at the S stage, my next little diagram. So this is just kind of your basic cell over here. It's one chromosome. You have your chromatid held by the centromere, and then you enter the actual S stage, which is when the after replication, the two sister chromatids are getting ready to split. Here they are genetically identical, again held by a centromere, and then next they'll actually move into the mitosis stage where they'll actually split. Okay, and that brings us to mitosis, which is when cells actually divide, and that helps them pass on genetic information to their daughter cells to carry out throughout our cells in our body. So to begin mitosis, we have prophase, which is when our chromosomes start to condense. We then have metaphase, which is when our chromosomes begin to line up in the center of the cell. We have anaphase, which is when the the chromatids actually split. We then go to telophase, which is when you start to kind of see that pinch off to actually make the two separate cells, but they don't actually split quite yet. And then finally, we have our cytokinesis, which is when the actual two cells split and you have two genetically identical cells that resemble what we had at the very beginning. Okay. And so continuing on, another subject we talked about was ALS, and how ALS is when nerve cells begin to die off. Usually symptoms are slurred speech or losing function of muscle movements. Once those neurons actually die off, then the muscles themselves will begin to die off. And neurons don't divide, so they can't regenerate after they die off because of the ALS disease. So one way that they are working on possibly curing this or helping this is by using stem cells, because as we know, stem cells can generate into many different types of stem cells. So the goal is to use stem cells to generate into neurons and then to use that as cell therapy for ALS people who are affected by that disease. And then finally, that brings us to our animal cells, which are eukaryotic cells. They have three major identifying factors. They do have a plasma membrane, which is just the boundary that lets things in and out of the cell. It has the nucleus, which stores all the genetic information, and then the cytoplasm, which allows movement within the cells. There are two different types of transportation in cells. You have passive, which doesn't require energy, and active. Looking at passive, which again doesn't use energy, you have diffusion, which is the random movement of molecules from areas of high concentration to low concentration. And you also have osmosis, which is the movement of water molecules from areas of high concentration to low concentration. And then that takes us to the active transportation, which does require energy, or ATP. So we do have these protein pumps that will allow movement from within with the help of ATP. And then you also have endo and exo cytosis, which allows movement either inside or outside of the cells with the help of ATP as well. And that is the basics to an animal cell.